thank all the speakers that have set the stage for this final talk today. It's been a very successful day, and I think we heard a lot about drug development. I'll try to put this in the framework of what we're trying to do at the Seaver Center to help patients with all forms of autism, including rare, monogenic forms, and idiopathic forms. So let's see if I can work this. Not so much. So we went over this paradigm many times today, talking about how lengthy and expensive and risky drug development is as an endeavor. And what we're trying to do here at Seaver is change this paradigm, offer a timeline that's going to be much abbreviated and much lower cost. And we hope that that new paradigm will be driven by the scientific knowledge that we have gained after 26 years of studying patients with autism and studying the very basic molecular processes at the core of the disease. As Jordi very eloquently elaborated on before, there is a set of specific challenges in neurodevelopmental disorders. Drug development is very challenging by its nature, but when you start to look at complex processes such as development, and you start to look at complex behaviors as humans tend to have, it becomes even more difficult to progress drugs through this process. We hope that by approaching this from an angle of monogenic disorders, monogenic forms of autism, we can simplify the process, and we can actually understand the biology at greater depth, which would help us progress drugs faster. We hope to improve the models that we use for screening new drugs. We talked a lot about stem cells and development of novel model systems that rely on stem cells. This is for the first time in the history of neurobiology that we can actually look at the tissue of relevance. We can look at the brain cells in a dish, real human cells, and understand not only what they look like, but how they connect to each other, how they talk to each other. We can understand that plasticity and build different models that would help us test therapeutic modalities in a relevant system. Modeling human behavior is not easy, especially in animals such as mice. As someone said before, we have cured many neurodevelopmental disorders in mice, but not so many yet in humans. We hope to take these animal models with a grain of salt and look at things that we can actually measure well and that we can translate into the human behavior, rather than try to mimic the complete human complexity in a much simpler organism. There is a huge diversity of neurodevelopmental disorders, even within the monogenic populations. As we see kids come to the Seaver Center day after day, each patient presents with his or her own set of challenges. And we hope that by observing the children as they develop and learning more about the symptomology of each individual patient, we can find common themes and we can find quantifiable aspects of their behavior. So we don't end up in a battle that Christine had to fight for her son and other boys with Duchenne. But we have some already relevant measures that we can look at and outcomes that actually matter to the patient's quality of life. And we really hope to bring forth our strong suit, which at Seaver is this integrative approach. We have a team of about 50 individuals looking at autism beginning to end, from very basic genetic discoveries to patients and complexity of human behavior. And we hope that by reiteration between these two ends of the spectrum, we can really get to the full understanding of the disease. And I will take you through the 4 specific aspects that create this spectrum of our activities. We can talk about the molecular targets that we're focused on, then the human neuron systems that we're developing, the animal models that we hope to use in screening of the novel therapeutics, and finally, and most importantly, about the patients and how we approach characterization of such a complex and diverse population. Joe had spoken about the discoveries that the Autism Sequencing Consortium has made over a number of years. This is by no means a small effort or a small feat. Very rarely will you see such a complete data set and such a large genetic data set. What the ASC has done, they have banded more than 40 international groups together, a huge number of researchers. They got over 36,000 genomes analyzed, and it all boiled down to 102 genes, which was kind of surprising because we always felt that something as complex as autism would yield many more genetic hits. But it turns out these are all the genes that we kind of already knew were involved. And we started to stratify them and look at those that really floated up to the surface. And Seaver Center has chosen to focus on some of them. Obviously, there are many other groups in the field working on other genes, but our current focus is, sorry about that, is basically these 4 that are circled in green or pink. So the green stands for our autosomal genes, SHANK3, ADMP, and FOXP1. And the pink is for girls with the DDX3X mutation, a relatively novel cause of autism in females. What makes these diseases and these genes of special interest to us is that they're monogenic, which means that a single mutation, even a point mutation in one of these genes, will lead to a full phenotype, to a neurodevelopmental delay and a behavioral phenotype that often falls under the umbrella of autism spectrum disorders. We see patients with all 4 of these disorders, and we're learning at the neck-breaking speed about the intricacies of the clinical presentation, and we're gathering data on the biomarkers. So having access to patients really makes this very attractive as a potential drug discovery effort. They are highly penetrable, which means that the child with a mutation in this particular gene will most likely have a disorder and have a syndrome. And we hope to move them all 4 ahead. You know, I have spoken to a lot of people since I joined Seaver Center, and everyone has their favorite gene. But my philosophy is that as we look at all 4 of them together, we'll be better able to implement lessons learned. And they will probably have different mechanisms. What we know about these 4 genes and their functions so far indicates that there will be very different. But certain tools that we're developing will be able to use across the board. Certain knowledge that we gain will be able to apply broadly. And we hope to move all 4 indications as fast as we can. And one of the very unique advantages that we have, and that comes from having access to the patients, is the ability to access patient samples and use their blood to develop stem cell models. And we have already been over the diagram on my left, showing basically how you can take a blood sample and you can reprogram the cells, generate iPSCs, and then program them into any specific cell type in the body that you want. Being that neurodevelopmental disorders come from neuronal function that's altered due to the genetic change, we would obviously reprogram cells into neurons or maybe supportive cells that are called glia, and then look at different types of culture in the dish and maybe build them into little mini organs or organoids. But what's important about these assays is that not only do we have access to the patients, not only do we have the knowledge on how to build these model systems in vitro, in the dish, but these assays have over time, thanks to the development of technology, become much more reliable. Not only can you use them in creative ways to understand the biology, but you can also use them for high throughput screening of different molecules. And you can see that the translatability and the quality of these assays has dramatically improved over the last 5 to 6 years. And they're becoming much more commonplace in discovery and development of psychiatric and neurological drugs. I want to draw your attention to the fact that we're building a little biobank from all 4 disorders that I mentioned before. We have a number of samples already collected, number of iPSC lines generated, both from patients and their unaffected siblings. We also combine that with the CRISPR-Cas approach that allows us to create the same mutation in the isogenic background. Stormy spoke a little bit about the advantage of having these identical twin cell lines when you can compare the normal genotype to the same genotype with a single mutation in it and derive conclusions based on the differences that you see in the cellular phenotype. So we have over 90 families that we collected blood from for these 4 disorders, and we have reprogrammed over 40 of them, and we hope to continue this initiative and to leverage this resource in the future drug discovery. We have already started looking at these cells, and we have several collaborations. Seaver Center, as big and as productive as it is, cannot handle as much work as we have to do here, so we collaborate with a lot of leaders in the field. This is collaboration that we have with Scripps Institute down in Florida. They have already developed an assay that looks at neurite outgrowth. This assay is a good measure of neurodevelopment in general. It's a very simple and straightforward assay that they can do really well and in a high throughput mode, which means that they can screen a relatively large number of compounds in a very short period of time. The cells that they use have been sort of characterized and proven to look like neurons, to behave to neurons as much as you can do that in a dish. And we hope to use this assay to look at the differences that are caused by deficit in these 4 genes and compare that to the relevant controls. We can do, as I said, this in many different ways, by looking at the isogenic lines or looking at the patient lines and comparing them to their sibling. We can even repair the genetic mutation in a patient and bring it back to the so-called wild-type state. And hopefully, once the phenotypes are established, we will be able to leverage this tool as a foray into drug discovery and look at some of the existing compound libraries to identify phenotypically what molecules could change the deficit in neurite outgrowth, and then take that into further characterization and to understand the actual mechanistic change that happens once you expose neurons to a specific compound. I'm sorry about that. Another assay that we're looking at that's also highly relevant, and this brings us a little closer to the actual function that we're trying to repair, is the optical electrophysiology. So in other words, neurons perform their function by communicating to each other, by firing these action potentials and transmitting signals through electrical conversion. And based on the phenotypes that we have established both in patients and also at the cellular level in 4 disorders I mentioned, we expect that there would be an electrophysiological phenotype, that there would be deficits in the way the neurons communicate with each other and the way they fire and their behavior in terms of the electrophysiology. So we're working with a company called Q-State in Cambridge. They have developed a state-of-the-art microscope that can be used to simultaneously measure electrophysiological parameters in hundreds of cells. This is a great improvement over how this method is conventionally used. When I first started dabbling into neuroscience many years ago, I sat in a dark room and patched the cell half an hour at a time, and I was so excited because you could see the cell firing, and at the time I thought that was the most wonderful experiment you can set up. But unfortunately, it only gains data from 1 cell in an hour. So you can't get very far by using just that approach. In order to screen large libraries, in order to understand physiology in a high throughput way, you really have to create this matrixed approach when you measure multiple parameters over multiple cells. And then we want to take this into another direction of complexity. So obviously, in the brain, neurons do not exist in 2-dimensional space, interacting with each other in a simplistic fashion. They get organized into different layers of the brain. They differentiate in different directions, in different neuronal subtypes. The novel approaches that allow us to build organoids, or as Jody calls them, mini brains, are something that we want to look at and deploy in the context of our 4 different types of cell lines. And we have partnered up with Rumi Scientific to develop an assay suitable. They already have an assay. We would like to further optimize it in the context of our cell lines and identify phenotypes that would potentially be screenable and targetable with new therapeutics. And then with the idea that once we find a therapeutic modality, we would have to take this down the path of complete preclinical discovery and development and then clinical development. We would like to tweak some of the models that have been built by the field and by the Seaver Center and Joe's lab for a long time. There is a number of animal models that we have with modified mouse genes, with human genes expressed in the mouse. What's lacking is really translatability. These models are relatively easy to manipulate in the lab, and there has been a long-standing history of positive data in the animal models followed by negative data in human trials. So what we're trying to do with the animal models is develop better outcomes, develop better measures that would actually help us understand the quantifiable characteristics of the models. So we're putting quite a bit of time and effort into designing experiments that would allow us to measure the same biomarkers in the animal models as our clinical team is measuring in the patients. We're looking in paradigms such as visual evoked potentials, auditory evoked potentials, and other EEG measures that are routinely implemented in the clinic. So that we can assess the impact of potential therapeutics on these measures in the animals, and in that way de-risk them for further development. This is not, you need to really approach this systematically. We hope to work with different leaders in the field, including companies such as Psychogenics. We have a close collaboration with Boston Children's. We're hoping to really, as a field, move this forward and establish a better battery of tests that we can use in mouse models and rat models of neurodevelopmental disorders. And another obvious thing that we have talked about during today's conference, and Stormy gave you a beautiful example of Angelman Syndrome and how deep understanding of science and the biology can really lead to targeted drug discovery, leaves a lot of room for stipulation in terms of our four disorders. We know that they're all monogenic. We know that they're all driven by mutations. In most cases, we feel like that we understand the molecular mechanism, that the haploinsufficiency in a given gene leads to the pathology. Even in the cases when there is what appears to be a dominant negative mutation, we can stipulate about the molecular mechanisms of that change. So that opens up space for targeted intervention at the level of a single gene, basically going after the missing product or going after the aberrant molecule and correcting for that function. Development drug development in neurodevelopmental disorders seems to be lagging behind. And I always quote Marc Tessier-Levine, who I remember reading his papers when I was in grad school, admiring his elegant work. He changed directions in career many times. At one point, he was the head of oncology at Roche. And when asked why he changed directions and went from a very esteemed neuroscientist into oncology, he said, because this is time for oncology, and this was maybe 15 years ago. Now we can make an impact, and neuroscience is next, but that time hasn't come yet. I think we're now at the point when neuroscience is breaking that barrier. We have much better tools, and this review is from 2013, showing that there is virtually no novel modalities in neuroscience and psychiatric disorders. I think that has already tremendously changed, and we'll see influx of novel therapies as we learn more about the way neurons work and as we develop better tools to model them. One of the obvious things that come to mind when we talk about targeted approach is gene therapy. If the gene is missing, the logical thinking would be to replace the missing copy and repair the function by introducing the missing copy. There are different ways of doing that. You can do it ex vivo and then bring that back into the organism, or you can deliver it directly to the organism through different delivery modes. We don't have much expertise or experience at the Seaver Center with gene therapy, but we're teaming up with some groups that have built a lot of knowledge and have done this successfully in other areas, in other disorders. There are currently 2 gene-approved therapies in the US and many more to come. We hope to use this approach at least to at first theoretically show that this type of therapy could be a viable option for monogenic disorders, monogenic neurodevelopmental disorders, and then hopefully bring this to the clinical readiness and test it in patients. There is a lot of challenges. There is the question of the dose effect. Too much of a correct version of the gene could also cause a phenotype. There are safety risks. There is a question of expression, localization. Do you have to restore the gene function in all of the cells, in all of the regions of the brain? These are all the questions that we don't have good answers to, but it's only through experimentation and to looking at length at how these constructs behave in different models that will come closer to the clinic and be able to successfully deliver these therapies in human patients. And I continue to be amazed at the complexity of the RNA regulation, because when I was in college, we still had this sort of and that back in the 1978, the splicing was discovered as this novel mechanism. Since I was in college until now, there has been a slew of discoveries in terms of RNA regulation. There has been new categories of molecules and new therapeutic modalities. We are going to very indiscriminately look across all the potential ways to modulate RNA transcription and RNA translation in order to adjust the expression levels of correct alleles of our genes of interest. And we're also going to look at other modalities that could potentially interact with RNA molecules and alter their expression levels or their translation rates to restore, again, the physiological state. There are in silico models and tools that you can use to screen existing compounds that we're utilizing already and discovering some potential hits. This is a publication from 2016 from Matt Disney's lab. We hope to use this tool and other tools in that same space, as well as novel approaches like the one that was described earlier by Stormy, to try to modulate the RNA-related mechanisms and basically restore the expression to where we hope the clinical presentation would be restored to normal as well. So I can skip this and go straight to the patients. As I said, Seaver Center has access to families not only with rare monogenic forms, but we also see a relatively large number of idiopathic autism patients. More than 200 families go through the center every year. The beauty of our approach is that they all go through a pretty standardized set of assessments that Dr. Kalavzan and his team have put together. This includes the behavioral tests, the medical assessments, as well as the biomarker assessments. A very talented group of researchers is working very hard to quantify and really optimize them. And this is some of the data that they have already generated in the space of visual evoked potentials and auditory evoked potentials, which I mentioned earlier. It seems that these biomarkers are relatively stable, at least in the rare orphan disease populations. And this is, from the drug discovery perspective, extremely attractive, because if we can look at this population in terms of one of these biomarkers as homogeneous, then we can really measure things consistently. Because when you're looking at a new drug, you're not just looking at how good your measure is in capturing behavior or reflecting the clinical outcome. You also have to understand how much room for improvement you have. Can you measure the change? It's much more important than can you quantify a measure really well. So it looks like some of these measures really offer a very good potential for the effect size. So we can measure the change and we can see the normalization that hopefully will show to correlate to the clinical outcomes and the overall improvement in quality of life. So one of the big initiatives that we have right now is to develop this toolkit that will help us go to the FDA hopefully one day and come prepared and say, look, we have these biomarkers, we have a patient population that's pretty stratified based on this. We can see the improvement in the biomarker. We have shown the correlation to the clinical outcome. And more importantly, we have shown that this matters to patients and their families. And that will put us in a position that we already have the answers to the questions that Christine and her organization had to answer. So in summary, obviously, I'm still in my honeymoon period here at the Seaver Center, but I do feel very strongly about our position in terms of ability to make the discoveries and to accelerate them down the drug development path. Obviously, we cannot do this in vacuum. We're a relatively small organization, and we're open to working with others who are interested in developing drugs in the neurodevelopmental space. We would like to position the monogenic forms of autism as sort of a proof of concept to show that we can really change these complex behaviors and make progress and make improvements to it. And then we would like to use lessons learned in the monogenic forms of autism to stratify more heterogeneous idiopathic population and test at least some of these therapeutic modalities in idiopathic patients. We will continue to develop our toolkit. We will continue with development of cellular models and animal models, gearing them both towards high throughput discovery, but also towards more high sensitivity, low throughput approach that will help us understand the biology, because without understanding mechanistic nature of the pathological processes and the intervention, even more importantly, you cannot place the drug safely in the patients, and that's what really matters. And we hope to continue to leverage these model systems to screen compounds and develop novel therapeutic modalities, and then bring these drugs to our patients and utilize the relationships that we have built with patient families and with patient organizations, family organizations, and foundations to work jointly on development of these programs. And with this, I will close and thank Seaver Foundation for generously supporting our work. I will thank Joe, who's been a wonderful mentor in the past 6 months, and I've learned much more than I have learned from many other mentors. And I want to thank the entire Seaver Autism Center team, who's welcomed me and have shown tremendous level of enthusiasm and openness to this relatively new initiative. And more than anyone else, I want to thank the patients and their brave families who bring their kids in and who will forge boldly the path to new therapeutics. Thank you.